Thank you very much, Dr. Walenga. It's a pleasure to be here, as always, as part of the NATF Forum, and I want to congratulate Sam and the organization for putting on such a important and educational forum, and I'm very proud to be part of it. These are my disclosures, but in talking about this particular topic today, I feel that in spite of the fact that I have an opportunity to work with any number of pharmaceutical companies, that I can present this topic in a very objective and unbiased manner. Well, on July 23rd of this year, the way that we do business in the United States as physicians who prescribe anticoagulation medications was changed dramatically. And that is the date that the FDA approved the Momenta version of enoxaparin as a follow-on biologic. And as I will discuss with you over the next 25 minutes, this is the first time that any biological product has ever been approved by the Food and Drug Administration in the United States without ever having that particular medication be placed into a human being before it was licensed for approval. So let's get some definitions formed so that we're all understanding what the issues are. First of all, what is a biosimilar or follow-on biologic? This is a formal definition by the Food and Drug Administration and should be compared to our concept of what a generic drug is. So the FDA says that a generic is a drug which is chemically based. However, a product is defined as biosimilar or a follow-on biologic if there's no clinically meaningful differences between the biologic product and the original reference product in terms of safety, efficacy, purity, potency of the product. So essentially then we're comparing the Momenta follow-on biologic enoxaparin to the branded enoxaparin already in the United States, approved by the FDA, made by Sanofi Aventis, called Lovenox. So what forces are powering this whole debate for the follow-on biologics? Well, I think that the bottom line here is that it's cost and cost alone. But it's also in the context of healthcare reform here in our country, the patent life, which has been extended in some legislation, but on many of these drugs is terminated over a very specific time frame. The technological advancements to be able to produce these follow-on biologics in a very efficient manner in the laboratory, although maybe not inexpensive, but much more effectively. And the fact that this is big business, and so there are many innovator and small companies out there who are interested in being able to get a quick process through the FDA in order to increase market share. And then this is a global precedent because essentially throughout the world there are many companies that are looking at the same issue of developing biosimilars in order to increase market share. So in the context in the United States, what is it that the FDA provides to us? Because as physicians, if we begin to administer certain pharmaceutical drugs, whether they're follow-on biologics or otherwise, we have the imprimatur of the FDA that allows us to give these medications, and we feel confident that the FDA has done everything that it can in order to provide us with indicators of safety and efficacy. Well, the FDA is supposed to protect public health and protect the pipeline of the drugs and how the drugs are manufactured in the manufacturing facility. It's supposed to provide advanced public health notices if there is a danger signal with any of the use of the medications that they have licensed. And the FDA is supposed to be very transparent in how it arrives at its decisions, and there are numerous websites now that you can see how the letters have been transmitted between the agency and the pharmaceutical companies, and how the deliberations have actually proceeded through the FDA. But cost has never been a mandate of the FDA. So if a drug looks good and it looks safe, it doesn't really matter what the cost of that drug is going to be. The marketplace will figure it out on its own. So the FDA in the United States is made up of 9 different centers. And the 2 centers that we're going to concentrate on today will be the Center for Biologics Evaluation and Research, so-called CDER, Biologics is CBER, and then the other center is Drug Evaluation and Research, which is CDER. Now, the interesting thing is that all of the biologicals used to be part of CBER. And then through a quirk of fate in 2003, the Office of Therapeutics that was in CBER was transformed over into the Center for Drug Evaluation. And part of that process took biologicals such as heparins and the low molecular weight heparins from CBER over to CDER. Now there's a big difference in the philosophies of how CBER approaches a biological versus how CDER approaches a biological. And CBER, as you can see, is responsible for many of the biologicals that are present in the marketplace now, whereas CDER has been mainly involved with drugs rather than with biologicals. So how does this decision tree actually work? So a company produces a drug, whether it's a follow-on biological or whether it's a generic chemical drug, and it sends it into the FDA, and then the process is determined whether or not the drug is going to go as a new drug or whether it's a different formulation on a previous drug. And in the 1980s, the Hatch-Waxman Health Reform Bill established new mechanisms to try to encourage the rapid transit, so to speak, of generic drugs through the FDA in order to facilitate the availability of previously very expensive on-patent drugs to the public as soon as the patent ended from many of these pharmaceuticals. So abbreviated new drug application pathways were established by the FDA. And 2 of these were labeled as 505B and 505J. Now 505B, as you can see, is a pathway through which clinical trials are going to be necessary. And you can see how there are different routes that you can arrive at this, but because of the interest of time, I'm not going to elaborate that much, except to indicate to you that the difference between these 2 abbreviated pathways, labeled 505B2 and 505J, were well delineated as far as generic drugs were concerned. That is, a generic Prozac, a generic antibiotic, etc. If the drug was a new formulation on the old drug, maybe a different dosing formulation, maybe the presence in a delivery system was different, then the 505J pathway was recommended because this abbreviated pathway allowed the pharmaceutical company to introduce a new drug without clinical trials. And in fact, the 505J pathway was so well delineated that the FDA could not even ask the drug company for any clinical data to show clinical efficacy and safety because it was presumed that that new construct was actually the same drug that the FDA had already approved in the past. If, on the other hand, the drug was somewhat different, then you would have to go through the 505(b)(2) pathway, and that required clinical trials, although on a smaller scale than what was originally required for the reference brand. As an example, Omnitrope serves as a way of discussing how these pathways were working for follow-on biologics. So essentially, this pathway was developed for generic chemical drugs, but not for follow-on biologics. And one of the first challenges for the follow-on biologics on these two pathways was Omnitrope, which is a follow-on biologic, genetically engineered human growth factor, which challenged the reference drug that was already on the market by Genentech. So the genetically engineered product, which was essentially the same as the Genentech drug, the company that manufactured that drug decided that it needed to have clinical trials. And interestingly enough, when it performed the clinical trials, there were pharmacodynamic differences between the Omnitrope, which was the follow-on biologic, versus the reference human growth factor from Genentech, indicating then that even though you have amino acid sequence identity, that even two drugs that could be characterized as being the same might have in vivo pharmacokinetic and pharmacodynamic differences. Well, there had not been any clear precedent for the use of the 505J pathway for follow-on biologics until the Momenta and enoxaparin story arrived. I think I'll skip this, except this is essentially a reiteration of what I was just talking to you about, that the genetically engineered product, even though it had biochemical identity, did not have in vivo pharmacodynamic identity. Well, at the same time that the FDA was looking at how to process follow-on biologics through various pathways, the European